Hi students, in this video, let's discuss about the chromosomal basis of inheritance. In the previous chapter, you have learned about Mendelian genetics. Now you are going to study with deviations of concepts related to Mendelian genetics and chromosomal theory of inheritance. Some of the key terms you must know in this chapter. What are autosomes? The body chromosomes are known as autosomes. So, any other chromosome other than sex chromosomes are known as autosomes. Then what is hemizygous? Having some single copies of genes. In the previous chapter, you have gone through about homozygous and heterozygous. Homozygous means a diploid organism in which both alleles are the same at the given gene locus is said to be homozygous. If it is present in different locus, then that is heterozygous. Then, here, what is wild type? The naturally occurring gene type is known as wild type gene. All the cells contain cytoplasm and nucleus. And inside the nucleus, chromatin reticulum is present. At the time of cell division, at the time of meiosis or mitosis, this chromatin reticulum uncoils and becomes chromosome. In the chromosomes, many genes are present, and each gene are present in specific locus. So, if you know all these basic things, you can understand easily the chromosomal theory of inheritance. The chromosomal theory of inheritance is the idea that genes, the units of heredity, are physical in nature and are found in the chromosomes. Chromosomes are seen in all dividing cells and are passed from generation to generation. Long before chromosomes were visualized under a microscope, the father of modern genetics, Gregor Johann Mendel, began studying heredity in 1843. With the improvement of microscopic techniques during the late 1800s, cell biologists could stain and visualize subcellular structures, the genes and the chromosomes. The non-blending inheritance was not at all proved by Gregor Johann Mendel, and he used mathematical calculations in biology, and no physical evidence of genes or chromosomes was submitted by Gregor Johann Mendel. So these are the reasons for non-acceptance of Mendel's work. The speculation that chromosomes might be the key to understanding heredity led several scientists to examine Mendel's publication and re-evaluate his model in terms of the behavior of chromosomes during meiosis and mitosis, during cell division. In 1902, Theodore Boveri observed that proper embryonic development of sea urchins doesn't occur unless chromosomes are present. That same year, Walter Sutton observed the separation of chromosomes into daughter cells during meiosis. Together, these observations led to the development of chromosomal theory of inheritance, which identified chromosomes as the genetic material responsible for Mendelian inheritance. Johann Mendel in 1865 studied the inheritance of well-defined characters. Three scientists, Hugo de Vries, Correns and Teschmark, independently rediscovered Mendel's results on the inheritance of characters. And Wilhelm Rox postulated chromosomes are responsible for transferring heredity. Montgomery concluded that maternal chromosome pair with paternal chromosome only during meiosis. T. Boveri, he proved that chromosomal theory of inheritance, and W. S. Sutton, a young American student, he recognized a parallelism between behavior of chromosome and Mendelian factors during gamete formation. Theodore Boveri was working on sea urchins, and Walter Sutton was working on grasshoppers. They both postulates the theory, the chromosomal theory of inheritance. And they proved individual genes are found at specific locations on a particular chromosome. The pattern of behavior of the chromosome during meiosis explains us the Mendelian laws as to why and how genes are inherited. Also, they proved the pattern of behavior of chromosomes. Thomas and Morgan, in 1910, he worked on fruit fly Drosophila melanogaster. The fruit fly completed their life cycle within 2 weeks. That's what he used Drosophila melanogaster for his experiments. And Drosophila, the gene for eye color is located on the X chromosome. Red eye color is wild type and is dominant, and the white eye color is recessive. The alleles for red or white color are present on the X chromosomes, but there is no counterpart for this gene on the Y chromosome. Thus, female have 2 alleles for this gene, whereas male have only 1. The genetic results were completely based on meiotic behavior of the X and Y chromosomes. This study strongly supports the idea that genes are located on chromosomes. Similarly, the genes for yellow body color and miniature wings are also carried on the chromosome. This linked genes connected together on sex chromosomes is called sex linkage. In this slide, they are given 4 diagrams of inheritance patterns of unlinked and linked genes. In the first diagram, 2 genes are located on different chromosomes, so independent assortment occurs during meiosis. The offsprings have an equal chance of being the parental type or a non-parental type. In the second diagram, 2 genes are very close together on the same chromosome so that no crossing over occurs between them. The genes are therefore always inherited together and all of the offsprings are the parental type. In the third diagram, 2 genes are far apart on the chromosome such that crossing over occurs during every meiotic event. During cell division, the recombination frequency will be the same as if the genes were on separate chromosomes. In the fourth diagram, the actual recombination frequency of fruit fly, wing length and body color that Thomas Morgan observed in 1912 was present. A crossover frequency between 0% and 50% indicates that the genes are on the same chromosomes and crossover occurs some of the time. This is the diagram explained in your textbook about the structure of somatic and sex chromosomes in Drosophila and sex linkage. So, what is sex linkage? Very important 2-mark question. Sex linkage is nothing but the linked genes connected together on sex chromosome. And it was explained by Thomas Hunt Morgan. Thomas Hunt Morgan in 1933 received Nobel Prize in Physiology or Medicine for his discoveries concerning the role played by chromosomes in heredity. The comparison between gene and chromosome behavior is given in your text. Around 20th century, cytologists established that generally the total number of chromosomes is constant in all cells of a species. And many organisms and number of chromosomes present in that organisms are also simultaneously given in your textbook. That in Ophioglossum, 1262 chromosomes are present. And Equisetum, horsetail, 216 chromosomes. And giant sequoia, 22 chromosomes. Then in sugarcane 80, apple 34, rice 24, potato 48, maize 20, onion 16. So like that, in human beings, there are 23 pairs of chromosomes. You know very well about that. And here, they compared with the gene and the chromosome behavior. A diploid eukaryotic cell has two diploid sets of chromosomes, one set from each parent. All somatic cells of an organism carry the same genetic complement. The behavior of chromosomes during meiosis not only explains Mendel's experiments but leads to new and different approaches to study about heredity. So, conclusion on comparison of chromosome and gene behavior is given in this tabular column. According to Mendelian factors, alleles of a factor occur in pair. Same as the chromosome behavior, chromosomes occur in pairs. Here, similar or dissimilar alleles of a factor separate during gamete formation. Here, homologous chromosomes separate during meiosis. So, Mendelian factors can assort independently. The paired chromosome can separate independently during meiosis, but the linked genes in the same chromosome normally do not assort independently. Was proved by Mendel. And it was hypothesized by Boveri and Sutton. So, the chromosomal theory of inheritance states that chromosomes and genes are present as pairs in diploid cells. Homologous chromosomes separate during meiosis. Fertilization restores chromosome number to diploid condition. The chromosomal theory of inheritance claims it is a chromosome that segregates and assort independently. The essence of important aspects of chromosome behavior in cell division is given in this slide. Alleles are found in the same locus. Locus means position on the chromosome, the gene position on the chromosome. Chromosome replicates in the S phase. Homologous chromosomes segregate in anaphase 1. Separation of sister chromatids in anaphase 2 are observed. So, the key points of chromosomal theory of inheritance are given in this slide. Homologous chromosome pairs are independent of other chromosome pairs. Chromosomes from each homologous pair are sorted randomly into pregametes. Parents synthesize gametes that contain only half of their chromosomes. Eggs and sperm have the same number of chromosomes. Gametic chromosomes combine during fertilization to produce offspring with the same chromosome number as their parents. Eye color in fruit flies was the first X-linked trait to be discovered. Thus, Morgan's experiments with fruit flies solidified the chromosomal theory of inheritance. Thank you, children.